We start from sex determination, so the actual chromosomes which determine the genetic sex of a person, and then we move on to sexual differentiation. So in other words, the actual development of the internal and the external sex organs of both males and females. Humans are sexually dimorphic. So this means that males and females are physically distinct. So although this distinction or this difference can often be blurred by dress, hairstyle, etc., etc., the physical distinctions are clear. So both male and female sex organs, although distinct, have or consist of three sets of structures, of which the first is the gonads. So the gonads are the organs that actually produce the gametes. So the male gonads are the testes, and the male gamete is the sperm or spermatozoa. And the female gonads are the ovaries, and the ovaries produce the female gamete, which is or are the eggs. So it's these gametes which then unite to form a new individual. The second set is the internal genitalia. So the internal genitalia basically consists of all the accessory glands and the ducts that connect these gonads, so these gamete producing organs, with the outside or the external environment. And then there are the external genitalia, so the external structures or the external reproductive structures. Each nucleated cell within the body, except for the gamete, so except for the eggs and the sperm, contain 46 chromosomes. So this set of chromosomes is called a diploid number because in actual fact these chromosomes occur in pairs. So these 46 chromosomes are actually 23 pairs of chromosomes, and these 23 pairs of chromosomes are then subdivided into 22 pairs or 44 chromosomes, which are somatic chromosomes or autosomal chromosomes. And these 22 pairs of chromosomes are the chromosomes which direct the development of variable characteristics. So in other words, general characteristics such as hair color, blood type, body form, etc. And then the other one pair that's left of the 23 pairs is one pair of sex chromosomes. So in other words, there are 2 sex chromosomes. And these sex chromosomes are the chromosomes which determine the gender. So they contain the genes which actually direct the development of the various sex organs into either male or female. So genetically, when there are 2 X chromosomes, it means that the person is genetically female, and an X and a Y chromosome refer to a genetically male person. Okay, and this figure just shows the 46 chromosomes, so the 23 pairs of chromosomes. So number 1 all the way through to number 22, those are the 22 pairs of autosomal chromosomes. And then over here is the 23rd pair, which consists of the 2 sex chromosomes. So according to this figure, if we look at these sex chromosomes, obviously this person would be male because there's an X and a Y chromosome. Okay, so what makes the gametes different, or the eggs and the sperm, different from all the other nucleated cells in the body? So remember we said that all of the other nucleated cells in the body have diploid numbers of chromosomes. So in other words, they have paired chromosomes. What makes the gametes different is the fact that they are haploid cells. So in other words, they have 23 chromosomes, not 23 pairs of chromosomes. So each egg and each sperm only has 22 somatic chromosomes and 1 sex chromosome. So in other words, when an egg and a sperm unite and form a zygote, a unique set of 46 chromosomes is then formed. So one set then comes from the mother and one set comes from the father to create this zygote with a unique set of DNA. So through a process of meiosis, cells go from a diploid number to a haploid number, so in other words containing only 1 sex chromosome, not one pair of sex chromosomes. So in female parents, the gametes or the eggs produced contain an X chromosome. And in male parents, the gametes or the sperm produced contain either an X or a Y chromosome. So females would inherit one X chromosome from the mother and one X chromosome from the father, whereas males would inherit the X chromosome from the mother and the Y chromosome from the father. So basically the sex chromosome, or the pair of sex chromosomes which a person inherits, determines the genetic sex of that individual. So after the fusion of the female egg and the male sperm and the formation of the zygote, the specific sex chromosomes within that zygote will direct the differentiation or the development of the sex organs of that zygote. So although the genetic sex is immediate, the actual differentiation of the three structures of the sex organs happens through time. So that only starts in about the 7th week of development. So in an early embryo, it's very difficult to determine the sex because the reproductive structures haven't actually started to differentiate. So this differentiation only starts by about week 7 of development, and it's only possible by about week 18 to 22 to find out the sex of the fetus. Now, before this differentiation takes place, all three structures, or all three sets of structures which we looked at in the first slide, which make up the sex organs, they are considered to be bipotential. So bipotential telling us they have a dual potential. So up until week 7, the different structures, the gonads, the internal genitalia, and the external genitalia, still have a dual ability to either become male or female structures. And all of this, the differentiation, is driven by the sex chromosomes. So the bipotential gonad consists of an outer cortex and an inner medulla. And under the influence of an appropriate developmental signal, the medulla will develop into the testes, and in the absence of this signal, the cortex will then differentiate into the ovarian tissue. So you see, depending on the sex chromosomes, either the cortex will develop or the medulla will develop. The bipotential internal genitalia consist of two pairs of accessory ducts, the Wolffian ducts and the Müllerian ducts. So as the development progresses along either a male or female pathway, one pair of these ducts will develop and another, whilst the other one will degenerate. And then the bipotential external genitalia consist of the genital tubercle, the urethral fold, urethral groove, and the labioscrotal swellings. So these different external structures will then differentiate into the male and the female reproductive structures as the development progresses. So what actually directs this single cell zygote to become either a male or a female fetus? So yes, we've said that the sex chromosomes that zygote has will drive the whole process of sexual differentiation, but it's actually even more specific than that. So this entire process of sex determination and sexual differentiation depends on the presence or absence of the sex determining region of the Y chromosome. This is also referred to as the SRY gene. So in the presence of this region or this specific gene, the bipotential gonads will develop into the testes. And in the absence of this gene, the bipotential gonads will then develop into the ovaries. So actually this whole process of sexual differentiation specifically depends on the presence or the absence of the Y chromosome. Okay, so we start off by looking at the specific differentiation or development of the internal genitalia. So when we say internal genitalia, in this picture we can see the bipotential gonads. So the gonads you can see over here. I don't want to color in the whole picture, but over here you can see the gonads, both of these. So the gonads you can see consist of an outer layer, that pinkish layer which you can see over here, the pinkish layer, the outer layer which is the cortex, and the inner layer, that's purple layer over here, which is the medulla. We can also see the bipotential ducts. So we have the Müllerian ducts over here, these purplish ducts, and these yellow ducts are the Wolffian ducts. So we say bipotential because this fetus is 6 weeks into development, which means that at this stage, all of the internal reproductive organs, so the gonads, as well as the rest of the internal genitalia, still have a potential to develop either into male or female structures. And we are going to start off by looking at the development of male structures. So in other words, in males, there is the presence of the Y chromosome. And when the Y chromosome is there, it means that the SRY gene is there. And when the SRY gene is there, it means that this gene produces SRY protein. And this SRY protein within the male embryo then directs the development of the medulla. So the internal purple part of the gonad is then directed to develop into the testis. So that is how the development of the male gonad is directed, by influence of the SRY protein. Now once the testes start to differentiate under the influence of the SRY protein, the testes then start to produce anti-Müllerian hormone, or AMH, anti-Müllerian hormone. So obviously AMH does exactly what its name says. It causes the Müllerian ducts to disappear. So the Müllerian ducts will then start to disintegrate, whilst the Wolffian ducts will be the dominant ducts to develop. Another hormone produced by the testes is testosterone, and testosterone plays a very important part in converting the Wolffian ducts into the male accessory glands and ducts. So in other words, into the seminal vesicles, the vas efferents, and the epididymis. And then also later during fetal development, it's testosterone which actually controls the migration of the testes from the abdomen into the scrotum, or the scrotal sac. But DHT, or dihydrotestosterone, which is a testosterone derivative, plays an important part in prostate development. And it also facilitates or plays an important part in the differentiation of the external genitalia. So testosterone, very important for the development of the internal genitalia, while DHT, or dihydrotestosterone, is important for prostate development and differentiation of external genitalia. So now we move on to the development of the female internal sex organs. So in the absence of the SRY protein, because we know that females are genetically composed of 2 X chromosomes, so there are no Y chromosomes. So without the Y chromosome, it means there's no SRY gene, and without the SRY gene, there's no SRY protein. So in the absence of this SRY protein, the cortex of the bipotential gonad, so the pink part of the bipotential gonad, will then develop into the ovarian tissue. So because it's ovarian tissue that is developing and not testes, it also means that there's no testosterone available. And without testosterone, the Wolffian ducts start to regress. Because if you remember from looking at the male development, it was testosterone which actually stimulated or converted the Wolffian ducts into the male accessory gland. So with no testosterone, the Wolffian ducts will then regress. Okay, and then also obviously with no testes that have developed, it also means that no anti-Müllerian hormone is being produced. So it means that the Müllerian duct actually remains, and the Müllerian ducts then start to develop into the female accessory structures and the internal genitalia of the female. So this includes the upper portion of the vagina, the uterus, the fallopian tubes, etc. And also, in the absence of DHT, so there's no dihydrotestosterone, it means that the external genitalia will then also take on female characteristics or start developing into female external genitalia. This just serves as a summary. So which parts of the bipotential sex organs develop into which parts of the male and female internal genitalia. So during male development or male differentiation, so when there's an XY chromosome, so in the presence of the Y chromosome, in the presence of the SRY gene, the cortex will regress while the medulla will develop into the testes. And because the testes have developed, it means that testosterone is present, and testosterone will cause the Wolffian ducts to then develop or produce the internal male genitalia and the male accessory structures. And also with the presence of the testes, it means that anti-Müllerian hormone is present, and the anti-Müllerian hormone will then cause the Müllerian ducts to regress. To the opposite end, during female differentiation, so when there are two X chromosomes, meaning that there is the absence of the Y chromosome, it means that the cortex will then develop into the ovary while the medulla regresses. And because there is no testes which develops, it means that there's no testosterone which is being produced, and there's also no anti-Müllerian hormone, which means that the Wolffian ducts will regress and the Müllerian ducts will develop into the female accessory structures and internal genitalia. Okay, so now we move away from the development of the internal genitalia and we take a look at the development of the external genitalia. So first of all, at the bipotential stage, so at age 6 weeks, during the fetus development, the fetus has bipotential external genitalia, and the bipotential external genitalia consist of the labioscrotal swellings, the genital tubercle, the urethral groove, and the urethral fold. So in other words, what we're saying is that at 6 weeks before development or differentiation of these external genitalia, it's not visually possible to identify this fetus as either male or female. But then, as we said, from about week 7 onwards, then the differentiation or the development begins. So first of all, we start off with differentiation of the male external genitalia. So remember, with the presence of the testes, we also have presence of DHT or dihydrotestosterone, which is that derivative of testosterone. And DHT is mainly responsible for the development of the male external genitalia. So it's dihydrotestosterone which actually controls the differentiation of the external genitalia to take on male characteristics. So it causes the masculinization of these bipotential external genitalia. And then it's only after birth in which the testosterone actually controls the migration of the testes from the abdomen into the scrotum or the scrotal sac. That only happens after birth. And this also happens due to the presence of testosterone which is produced by the testes. And then to the opposite end, in the absence of all of the male hormones, all of the male androgens, the bipotential external genitalia, as seen in the picture, are then feminized. So in other words, the bipotential external genitalia take on female form, and they then become all the female external genitalia. Okay, and just a summary of the development or differentiation of the external genitalia. So during the external development, all of these external bipotential structures will then develop into either male or female sex organ. So during male differentiation of the external genitalia, the genital tubercle will become the glans penis. The urethral folds and grooves, they will become the shaft of the penis, and the labioscrotal swellings will become the scrotum and also form part of the penis. During female differentiation of the external genitalia, the genital tubercle forms the clitoris. The urethral folds and grooves, they then become the labia minora, the opening of the vagina and the urethra, and the labioscrotal swellings, they then become the labia majora. Okay, so in actual fact, it's under the direction or the presence of the SRY gene that male development or differentiation is then driven. Obviously, under the absence of this SRY protein and gene, and under the direction of multiple female-specific genes, the gonads would develop into the ovaries. But with the presence of the sex-determining region of the Y chromosome, or the SRY gene, this leads to the production of the SRY protein, and this leads to the development of the gonad into, specifically the medulla of the gonad, into the testes. The testes then have two cell types, the Leydig cells and the Sertoli cells, and it's the Leydig cells which lead to the secretion of testosterone, and testosterone causes the development of the Wolffian ducts into all of the accessory structures, and then indirectly it's the testosterone which causes the development of the external genitalia. We say indirectly because it's actually DHT or dihydrotestosterone which is directly responsible for the development of the external genitalia. But DHT is a derivative of testosterone. So testosterone becomes DHT by 5-alpha reductase, an enzymatic reaction. Testosterone is converted into DHT. Then it's the Sertoli cells which secrete the anti-Müllerian hormone, and of course it's the anti-Müllerian hormone which then causes the regression of the Müllerian ducts. Okay, so now we know how male and female gonads differentiate or develop. And now we move on to take a closer look at the production of gametes or gametogenesis, because remember we said it's the gonads which actually produce the gametes. Now, the first step of gametogenesis is mitosis. So during this process of mitosis, there is cell division which is taking place. Specifically, division of the germ cells. So there is germ cell proliferation. So what are germ cells? Germ cells are the cells within the gonad which are actually destined to produce the gametes. So in other words, the eggs or the sperm. And during this first step of proliferation, each daughter cell which is produced is identical to the parent cell. So here we have an oogonium, one oogonium, so that is the singular form, and a series of mitotic divisions take place to produce many oogonia, and the same happens on the male side. So the daughter cells each have exact genetic composition as compared to the parent cell. So basically what's happening now is in both sexes, the germ cells within the embryonic gonads are undergoing the series of mitotic divisions in order to increase their numbers. After this first stage of mitosis, the germ cells are then ready to undergo meiosis. Now, meiosis is actually the cell division process that forms the gametes. Now, the first step of meiosis is when the germ cells actually duplicate their DNA. So each chromosome is duplicated so that they form these sister chromatids. So you can see there's duplication of all of the DNA over here. Okay, so there's the formation of these sister chromatids. So these cells that have now been formed during the first step of meiosis are referred to as primary oocytes or primary spermatocytes. So it's not germ cells anymore. The oogonia have now become primary oocytes, and the spermatogonia have now become primary spermatocytes. Now these primary oocytes or spermatocytes contain twice the normal amount of DNA. So no cell division has taken place, but there has been replication of the DNA. So there are 46 chromosomes still, because that's the diploid number they started off with after the first mitosis process. But now these 46 chromosomes have been duplicated into sister chromatids. The next step in meiosis is the first meiotic division. And during this division, the primary gamete, so the primary oocyte and the primary spermatocyte, divide into secondary gametes. So a primary oocyte becomes a secondary oocyte, and a primary spermatocyte becomes a secondary spermatocyte. Now in females, the primary oocyte is divided into a secondary oocyte as well as the first polar body. And in males, the primary spermatocyte forms two secondary spermatocytes. So there's a difference at this stage of the meiotic division. Now each of these secondary gametes that are formed, the one that's formed in females and the two that's formed in males, they contain one copy of the duplicated chromosomes. So as you can see, one copy of the two sister chromatids, which are then linked at a centromere. During the second meiotic division, the secondary gamete divides. So there's separation of the sister chromatids, and the aim is to then develop a haploid gamete. So in males, the secondary spermatocyte, each one of these secondary spermatocytes develop or divide into spermatids. You can see that these sister chromatids have divided, so from two sister crids which are linked at a central centromere. The daughter cells, or the spermatids, which then form from these secondary spermatocytes are haploid. So they only have one sister chromatid, so in other words, a haploid number of DNA. So each secondary spermatocyte produces two spermatids. Now these spermatids then mature and develop into the actual sperm, or the spermatozoa, which are just flagellated. So if we look at this differently, we can say that each secondary spermatocyte develops into two spermatozoa, or two gametes. And if we take this even further back, each primary spermatocyte forms four gametes, or four spermatozoa. However, if we look on the female side, things are a little bit different. So in females, the second meiotic division, so the division of the secondary gamete or the secondary oocyte, results in the separation of the sister chromatids, but not the division of the actual oocyte. So this is what the ovum looks like, which is released during ovulation. So during the menstrual cycle, at around about day 14, this is what the egg or the ovum looks like, which is released. Now, what is shown here is only the first part of the second meiotic division. So there's only separation of the sister chromatids, but there's no division of the cell. And then there is disintegration of the polar body that was formed after the first meiotic division. So for females, it's different because one primary oocyte results in one egg or one ovum which is released. And remember, when this ovum is released, the second meiotic division has not yet completed. And in actual fact, if fertilization of this egg does not take place, and this egg will never undergo meiosis in completion. So if this egg is not fertilized, meiosis will never complete, So this unfertilized egg will just be passed out of the body in the state that it is in currently. However, if fertilization takes place by means of a sperm, so when that egg meets a sperm and fertilization takes place to form a zygote, then only does the final step of meiosis take place. So as you can see, half of the sister chromatids are then fused with the sperm to form the zygote, and the other half is then released in a second polar body, a smaller polar body, which is then released and disintegrated. So only once fertilization takes place, then only will meiosis, or the second division of meiosis, resume completely within the female reproductive system. Up until this stage, we were looking at the process of gametogenesis by specifically focusing on the cell divisions, the processes of mitosis going into meiosis, etc., as well as how many primary gametes form the final gametes, how many secondary gametes form the final gametes, those types of things. But for now, I want us to concentrate on the timelines, because timelines in both females and males differs for when mitosis and meiosis is taking place. So if we take a look on the male side, during spermatogenesis, we see that at birth, so this is during the embryonic phase up until there. So let's take birth as this point over here. So at birth, the testes of the boy has not yet progressed past mitosis. We can see that mitosis is still the process which is taking here. We're still in the phase of mitosis. So in other words, at birth, the male gametes are the spermatogonia, or the germ cells. And then after birth, there's a phase during which these germ cells, or the spermatogonia, become relatively inactive or quiescent. And then only until puberty does mitosis then resume. So during the adult reproductive phase, which we can see from here, so we take that as the onset of puberty and this whole part as the rest of the adult reproductive phase. So only from puberty does the mitosis of these germ cells continue and then carry on into meiosis through the formation of the gametes. Now this is very different in females, because if we look on the female side, if we take this part again as the embryonic development and this point as birth, then it means that mitosis of the germ cells have completed during the embryonic development. And in actual fact, these germ cells have already entered the first phase of meiosis. So in other words, the phase where the DNA replicates. So that means that at birth, females are born with the total number of primary oocytes they will ever have. So there's no further mitosis taking place after birth, which means that the number of primary oocytes that are present within the ovaries at birth, that is the number that the females will have throughout the rest of their lives. And this is usually about 500,000 or 500,000 primary gametes or primary oocytes per ovary. Then from the onset of puberty, during the reproductive adult phase, that is when meiosis then resumes from the first meiotic division onwards. And this part is the part that takes place during puberty, specifically the release or the ovulation, which takes place monthly by means of ovulation. So each month during ovulation, when the egg is released, it means that there is a reduction of the actual pool of primary oocytes available, which is opposite to the spermatogenesis on the male side, because sperm or spermatozoa can be endlessly reproduced on the male side, because mitosis still takes place during the reproductive adult phase, whereas mitosis has ceased completely during embryonic development or after embryonic development. So during the adult reproductive phase, there can be no increase in the number of the primary oocytes. Okay, and now we move on to the hormonal control of reproduction, which in both males and females follows the basic hypothalamic anterior pituitary peripheral gland axis, so the axis that we're quite used to by now. So in other words, there's stimuli which comes from the external environment. The hypothalamus is then stimulated to release gonadotropin releasing hormone, and this gonadotropin releasing hormone then controls the release from the anterior pituitary of the two gonadotropins, which is luteinizing hormone and follicle stimulating hormone. Both of these gonadotropins, the LH as well as the FSH, they act on the gonads. Now, the FSH specifically is required to initiate and maintain the gametogenesis process. And to the other end, the LH acts primarily on the endocrine cells of the gonads, stimulating them to produce the steroid sex hormone. So the testosterone in males and the estrogens in females. Luteinizing hormone does, however, contribute to gamete production in females as well. And then again, as with any endocrine pathway, there are feedback loops in place, both long loop and short loop negative feedback loops. But as you'll see later during the lectures that we do in reproduction, the feedback loops in the reproductive system are a little bit more complex. These feedbacks can be either negative or positive. This table basically shortly summarizes what I meant with the complex feedback loops. So different levels of the different steroid hormones actually affects the feedback on the gonadotropin release. So when the circulating levels or concentrations of the steroid hormones, so if estrogen and androgen is low, when the circulating levels are low, then there's absence of negative feedback, which is normal, which means that the gonadotropin levels, so the FSH and the LH levels, will increase. Now, as the steroid concentrations moderately increase, so as estrogen and androgen levels increase, this is when negative feedback is introduced. So this is still quite normal, which means that the negative feedback will then decrease the release of the FSH and LH. Now, high androgen concentrations always maintain a negative feedback on the gonadotropin release. So gonadotropin levels will always be decreased in light of high circulating androgen concentrations. However, this is not the case with estrogen. So this part is where the difference comes in. So up until here, the feedback loops are pretty normal and what we are used to in the other endocrine loops that we've looked at. But with sustained estrogen levels, that's where the difference comes in. So when estrogen rises rapidly and it reaches a certain threshold level and it remains that high, so at least about 36 hours, the feedback then switches from negative to positive for the estrogens. So gonadotropin release in the presence of sustained high estrogen concentrations, the gonadotropin release is increased. So FSH and LH levels will increase. So in other words, estrogen has this paradoxical effect on the gonadotropin release. And this effect of estrogen on the FSH and LH release plays a very important role in the female reproductive cycle or in the menstrual cycle, as we'll see when we take a closer look at the female reproductive system.